to the Weekly Bioanalysis, a KCAS podcast.
Hello, and welcome to the 55th episode of the Weekly Bioanalysis, the official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is Dominic Guarino, and I've been in the bioanalytical, LBA, and biomarker space for over 28 years. I'm here with my co-host, John Perkins.
Hello, my name is John Perkins, and I've been in the bioanalytical LCMS space for over 27 years.
John and I are senior scientific advisors at KCAS, and either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. I'm located in Kansas. John is in New York. Our producer is in Missouri. So we're kind of all over the map here. We are thrilled to have you visiting and listening to the 55th episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcast, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining for the first time or if you've been a regular. So, as many of you know, we started this podcast due to COVID. But now that those are, because of COVID, we were restricted with our continuing business as usual. So we weren't able to meet face to face. So that's kind of undoing. But throughout all of that, the podcast just became something that's part of KCAS. So we're going to keep doing it despite maybe COVID restrictions being lifted. If you're unfamiliar with our podcast, we will do a review of the latest news and resources and then focus on a topic of our choosing before discussing any feedback we've had from you. We're constantly looking for topics, and we'd be happy to discuss something that you may want us to cover. Then we'll wrap up and give you a little teaser for the next episode, letting you know what might be coming in the future. So again, we're ecstatic to have you here, and we are looking forward to a fun and exciting episode. John, why don't you give us a little bit of review about what we'll be going over today?
Sure. As usual, we'll talk about some recent news topics that have tickled our fancy. We'll start with some COVID items, moving into non-COVID. And then our main topic, we thought, as a change, we talk about the industry as a whole, but we thought we'd like to focus on KCS itself. We're in exciting times as a company, and we really want to talk about some of the investments and changes that are coming with us and where we're going for the future.
Yeah, John, we're a jam-packed episode of what we're doing here at KCS, the future and the investments that we're making. But with that, John, why don't you start us off with the news?
Start off with a couple of COVID items, which should be brief before we move into some more interesting topics. But the first one is FDA cuts off use of GSK's COVID treatment in almost half of the US as the BA2 variant spreads. So obviously, as we're still not really at that level where we're vaccinated enough, where we're preventing the mutation and spread of new variants. So we're still having to deal with these variants, and the latest of concern is BA2, which I think was somewhat related to Omicron. But the GSK and Vir sotrovimab, which was shown to be effective against Omicron, is now ineffective against BA2, and so it's a number of as soon as the BA2 is accounting for 50% of cases in states, then it's been struck off. The sotrovimab has been struck off the list of available medicines, and so it follows in the path of the Lilly antibody cocktail that was pulled after Omicron and the Regeneron cocktail too. So it's this constant battle between us and the virus.
Yeah, I think, the use of biologics, monoclonal antibodies was and still is a very powerful tool in the tool chest. It's just, you wonder if some of this is hopefully a waning, maybe, but clearly this cocktail mixture didn't work. But I still think biologics and monoclonals are going to play a critical part going forward with, therapies and treatments and having other things available so that we're not all...
I agree with you on that. I mean, it's. I think it's. I'm still see out there. COVID fatigue and rolling back of precautions, and I know at least in the UK, I've seen friends on Facebook. After 2 years being COVID free, announcing that I've tested positive. One of my friends, it was his birthday. a week ago, and on the day of his birthday said, "Yep, thanks for all the birthday wishes. Just tested positive for COVID." You know, and then this 2 or 3 weeks ago here, the school had every they always do like school plays and stuff, so they did a musical. And then there was a local outbreak that had obviously was really based around the musical at the school. So yeah, you still have to be cautious.
Yeah, John, we haven't had a lot of outbreaks here in Kansas. It's been pretty quiet in that respect. There's still cases, but they're very low. I think it's hopefully this herd immunity, right? Like at this stage, I hope a lot of it is people are getting sick and they're not, well, at least getting maybe COVID positive and they're not getting, hospitalized because it was so new to the environment, right? So I hope all of that is part of it. But yeah, no.
I think that's definitely the case. That when I talk about friends testing positive, they're not getting major symptoms. It's just it's like they're vaccinated and that's helping, but they are definitely still catching it. And actually
Yeah.
here, they're rolling out the second booster for people over 50. So there's still that second booster shot availability.
They're talking about a third and a fourth now, and I think if you're of a certain age, you should listen to your physician and take that advice. But and I think if you're of a other ages, maybe again listen to your physician. But I feel like this is these particular monoclonals are still going to play a major role going forward. I think the Regeneron cocktail is still a very good one, and more importantly, I think it's really advanced the idea of multiple multiple monoclonals just in therapeutics as well. And there's still that camaraderie, or at least that collaboration, I think is something that will continue, hopefully, to serve things like immuno-oncology and orphan diseases and stuff, not just COVID. But that's really the bottom line. I know it didn't work here, but sometimes, remember, John, we're scientists, and you learn more from what doesn't work than what does frequently.
And actually, there still are two antibodies that are authorized for use. There's a cocktail from AstraZeneca, which is Tizakevimab, Silgavimab, and then Lilly has another one, but sadly my printer died and I can't read the name of it. But there's
it's been Tulimab.
Yeah, so although some antibodies have been rendered ineffective, others are still being developed. So we're still keeping that.
And John, it's like convalescent antibodies or plasma has been determined as not effective, but I still think there's a role for that in certain individuals. It's just that 50% metric, doesn't necessarily mean it's just ineffective, right? It's not as protective or beneficial across a wider range. So it is what it is, and we'll keep reporting on therapeutics. But I think to our audience, even though there's a little bit of it appears not to be as effective as they like. But I don't think there's a lot of paranoia. Like you said, there's a couple cocktails or at least antibodies are available, and every day we're learning about more therapeutics that are.
Oh, sure. There's so many. In preparing for this, there's so many therapeutics in the pipeline that could contribute.
Ultimately, we hope we don't have to use them, right? Exactly. That's hope is not a plan, John. Right? That's the problem there.
So let's move on. So we've obviously talked about the Moderna vaccine, but Moderna debuts friendly Meet Spikevax animated video to explain how its COVID-19 vaccine works. I just thought it was worth highlighting this. It's a short 4-minute video, of which half of which is disclaimers, but it's actually it's quite a fun way it's presented. It talks about how mRNA works, rather it's not DNA, and it's just mechanism of action. It's really simplified for anyone to understand. If you have time, it's worth jumping on YouTube and just checking that out.
Jeremy, maybe we can get that out to. Oh, I see it, and maybe we can, when you post it, Jeremy. Let's try to get that out to the audience. I think that's a great educational tool, and everybody likes a cartoon, even if it's got a lot of disclaimers in it, right, John? It's good stuff.
Yeah, I happily sat through it. It was good.
Yeah, so, I don't know. Moderna certainly is. In this case, this is great, the video, but I'll take a moment. They're really ramping up, right? I mean, not just in the COVID space. They're constantly making the news in many ways, and one of the great advancements in mRNA technologies and in general gene therapies is due to COVID, and I believe they're expanding into multiple areas, including infectious disease and immuno-oncology. So more to see from Moderna, right?
I think and we'll talk touch on what COVID has done for the medical communities in terms of challenges and benefits. But really, for Moderna, it's proof of concept of their technology.
Yeah.
And so it's, they've shown effectiveness, and now it's then what other diseases can this technology then help and
Yeah.
guard against. So
And it's great. I want to emphasize to the audience, it's not new. It didn't happen in just
yeah.
like 2020. These
And
things are from the 2010s or early 2000s, right?
yeah,
So this is not new stuff.
their cartoon emphasizes that.
Yeah,
It
great.
does touch on that. Sure, so we move on to
Yeah,
next
yeah, yeah.
topic. This so obviously one thing that we've discussed and we touched on it, but COVID, obviously, we talk about friends who've had symptoms and then a week later we're all through it, etc. But there's a significant subset of the population who are suffering from long COVID, and they all present a mix of various symptoms, and we don't have a solution for that right now. So some people have been struggling with symptoms for months with no real end in sight. And there's a couple of articles. One's Big Pharma skirted long COVID, but a pipeline of small biotechs is testing treatments for lingering symptoms. And the other one is how long COVID is accelerating a revolution in medical research. And really, the gist of these two articles, and there's more out there, but we really, in terms of the high profile pharma, have focused on really addressing the basic trying to prevent COVID, and not trying to address these people who are dealing with the long term effects.
Yeah.
And so then the second part of it is because there's such a myriad of symptoms, even those researchers who are trying to figure out long COVID, there's no precedent for them. It's really difficult to understand what's going on. So they're really relying on patient advocates and patients themselves to record symptoms, progression, etc., so they can try and get a handle on long COVID. So it's like slowly trying to build the understanding of what's going in there, and patients are integral to it all.
Yeah, John, I think even as we were sitting here, I thought, wow, I do know somebody who had what is probably considered long COVID, right? And so that's still something that, and the numbers are staggering. They don't know. It's like 7.7 million to
Yeah,
upwards of 23 million, right? That's
yeah,
not a small delta. And I think it is underserved. There are and really it kind of speaks to the people who are addressing it are the ones that had it, right? Like researchers who got long COVID are the ones who are, but I think that is something that isn't really fully understood, and certainly isn't something that's making a lot of headlines, meaning an individual, if you get COVID, there's a large portion of them that have very mild symptoms, and within say 5 days they're remedied. And then there are others who suffer. There are some who are still sick for a couple weeks at a time, and then they have some lingering, whatever it might be, smell, right? They have some neurological
yeah,
things.
sure,
Those are
yeah,
what the long COVID falls in. That and interesting here. They need to stratify that, is how I think it. Meaning, there's got to be some connection between those people that hasn't been unearthed yet. Right now, it seems like it's a hodgepodge of things,
yeah.
but somewhere in there has to be some underlying thing that's causing those individuals. And John, it's a very hard thing to probably untangle, and it's going to take time, because it could be as simple as, gee, you just got a big bolus of COVID from somebody who didn't pass along any immunity. Who knows? To as simple, as complex as, well, whatever your genetic makeup is, whatever your, maybe some underlying health conditions that we don't know about, but whatever the reason is, it seems like it is striking individuals. And, unfortunately, I wonder if those individuals, there's a silver lining. Maybe through time it'll show, hey, you're maybe prone to X, Y, and Z because of this. I don't know, but I think there's a lot of things that the genes, right, are playing a big part in this.
No,
So
I mean,
it's fascinating.
Yeah, I have to wonder if there's some genetic
Yeah.
predisposition in some of these people.
Well,
But
we
I mean,
talk about it, right? The ACE2 receptors could be one, and hormone levels. Those are both things that are going to impact how you're going to deal with COVID and what the symptoms and then the ailment itself would be. Those two things are understudied, in my opinion.
sure, yeah. But I mean, there is a wide range of companies looking at this. They are smaller biotechs, and again, but not all the focuses are the same, because the long COVID people are, some are focused on the GI tract, Others are focused on it's this. I mean, some people have had
Neurology, right? Yeah.
yeah,
Sorry, I
some,
didn't mean to talk
yeah,
over you. I think it's hard because it's a wide net and it's a big mountain. It's like Everest. If I'm a biotech and I was an investor and you said, "Oh, I want to go after like long COVID," I'd say, "Wow, that there's a lot of other things we could go after." I think.
yeah, and it's also, I mean, but which piece? It's like, I mean, you've got people you read about people who've been suffering brain fog, that and that lack of clarity for months, and it's like, well, how do you drill into that? I mean, it's a big challenge, definitely.
Yeah, I mean, we've been searching neurodegenerative diseases for decades, and really, there's nothing on the market yet. Now
Sure.
we've got
Yeah.
some things that we think are good, but why? Like you just said, brain fog. Now we've got these new sort of disorders, and we can't remedy the ones we already have. So, the neurological space is probably the area that, hopefully our children, our grandchildren will benefit from all we're doing now. But it's just been very tough sledding to
Yep.
get through the
Yep.
neurodegenerative
Yeah.
space.
Quite the medical graveyard, really, in terms of companies who've tried and just not really made.
Yeah, and that sounds so morbid. There's been some successes, but not at a rate that would be greater than 50 or even above standard of care to some respect,
Yeah.
right? So that's
Yeah.
the challenge. But let's move on, John. That was a good one.
So yeah, this was a good sort of summary. And again, we can go through this relatively quickly, but it was 10 ways COVID-19 rocked biotech. And this was a fierce biotech summary, and some of it I didn't necessarily think about. But we're dealing with COVID. You're in the middle of it, and sometimes it does mean they did like a summary of 10 things, and a couple of them really were. Oh, I never thought of that. Yeah, that's
Mm.
really quite interesting. So we can pop through this and maybe just go through them one at a time, and some of them will be very quick. I mean, so the first one they cited was RSV, which is respiratory syncytial virus, and if you read like the medical, like these newsletters, RSV virus comes up quite often, as there were a number of vaccines in development already prior to COVID, and Sanofi in particular had one that was in the late stage of clinical development, but it got to stage if COVID was transmitted via respiratory mechanism, they couldn't, in all honesty, ask
Mm.
volunteers to go out and risk infection for them to prove the efficacy of the RSV vaccine, because they could get COVID and while doing that. So they pretty much had to kill their study
Mm.
stone dead. So at some point, obviously we'll pivot back to RSV, and there is a lot of attention on it. But it's just these are the sort of weird effects that this pandemic has had on research.
Yeah, there was a lack of people to study, right? So no
Yeah.
one was getting RSV infections because no one was doing anything, and so the trials got interesting.
So,
Um,
moving on, there's like, so obviously we've seen a bunch of COVID focused biotechs. A number of those were already working on like SARS type viruses anyway, and they pivoted to COVID. But it's also because things with COVID have moved so quickly, it's actually chewed up and spat out biotechs quite quickly as well. There was the one of the government funded studies was shut down with a couple of biotechs who had products that were moving forward, and they just went no further, and they're sort of now left hanging.
yeah,
Um, we
interesting. No, I think I hope that makes a lot of sense to everybody. New biotechs were focused on COVID, and that's just going to be volatile as
Sure,
that
absolutely,
market
yeah,
is.
yeah.
And there's been a lot of volatility in general in the biotech and pharma space, but I think it ballooned so much, and that's a different topic, right? There's
Sure,
still a lot.
yeah, and we're
Anyhow, let's move on to. I think this is an interesting one. The decentralized trials take root, which
so. Do you want to do the other one first, since you touched on it earlier? Obviously, with the vaccines, BioNTech and Moderna have become household names.
yeah,
No, so we sort of, I think we were aware of Moderna, but I think BioNTech have really been a name that has appeared through the partnership with Pfizer, and so now they've obviously made money on this. So now it's where they're looking to reinvest. So we talked about Moderna really looking to, it's proof of their technology now, looking to apply those vaccines to other things. BioNTech is doing a similar thing. They're working now on a shingles vaccine to possibly go up against GSK's Shingrix, which we view as point of care with as a good preventative vaccine
Yeah,
for
it's
shingles.
it's a standard of care. It's recommended for anybody over the age of 50. Yeah. No, interesting. You're right. There's a lot of good, the BioNTech and Moderna are now up there with Biogen's, Regeneron, Takeda type names. So they're interesting stuff. And then I think BioNTech has immunotherapy coming, right? So that's another, kind of,
Yes.
I guess it's good. Good comes to mind, right? More
Yep.
better. Yeah. Yeah, John. So and thanks for taking a step back. And it is important to note that one of the big outcomes of COVID is we've popped up some what we'll call superstars in the space, and BioNTech and Moderna lead that space for sure.
So you can go with the decentralized trials now, because
Yeah,
this changed how trials are run.
I think it's pretty interesting that now it seems like with all this we because of COVID, we're now allowing people to what's called decentralizing trials, and that still doesn't resonate well with me. But John, that's just the way of the world, right? So that
Yep.
means less people going into a clinic and people doing it more online, right? So is that how you view this, or did I miss the mark?
I mean, to me, I think it's sampling off site. I think a lot of discussion. What is not clear to me is where samples are taken. Is that at the clinic or do they do it at home? But certainly a lot of the information in terms of registering people, etc., that can all be done from home. I assume
Yeah,
that's
and that's what was unclear to me, right?
yeah.
Is it just as simple as, oh, you're taking your blood pressure and your temperature, or are you actually at home, like, I don't know, doing a finger stick and sending it off or trying to draw? I couldn't quite tell what that meant, but clearly I know there's been a lot of information and what enrollments and things like that are no longer requiring you to go into the building, right?
Yeah,
So,
so
it,
so
yeah,
it changes how the trial is put together, and honestly, once you go down the one step towards this, there'll be a continued evolution in terms of how our trials carried out, particularly the late stage ones where it's hundreds of patients.
yeah, I guess there's still that challenge of getting a sample of blood, a couple mils of blood, which to me, I don't know how they're going to overcome that. Maybe it's more, but it's always you don't always have to go to a phase one unit or some sort of hospital. There's been other offshoots. There's a lot of like little. If you ever drive around, you see like a little lab corp sign on something.
Sure.
That's a
Yeah.
place where someone can go and get their blood drawn, right? So, but interesting. I do think
And
I think the take home is there's been a massive change, even a revolution in clinical trials due to COVID.
Sure, and I mean, for example, there is focus on within the bioanalytical space. How can you possibly sample patients at home? Where I mean, there's a couple of companies produce these, particularly microsampling driven,
Mhm.
where it's a thing that you wear on your arm, and it will then at a certain time you know
Makes sense.
it
Yeah.
take a microsample, and I know, for example, Merck were very interested in how that could be
Well,
used.
as a sidebar, that all comes from the diabetic space, right? There's been the people who have those. I mean, diabetic pumps have been what a wonderful advancement in science in our lifetime, John. Right? So they've been able to. They used to be like somebody had to carry like a 5 pound thing with them to be. Now they just slap it on them. These small little pads, essentially. And like you said, it's a micro sampling system, so that could evolve into what they're using here, I would think, right? So
Yeah,
that
yeah,
could be
yeah.
the sort of way to it's a little bit different measuring insulin or A1C versus measuring, say, a biologic or a small molecule, but don't put it past anybody to come up with a way to make it smaller and better, right? So,
And yeah, because obviously and bioanalysis has evolved so quickly in the last 30, 40 years. If there's something like that out there that can prove useful, it will prove useful,
yeah.
and that'll be the
It'd
direction.
be like, well, lateral flows will give you pluses and minuses, but they won't give you like PK profiles, right? So that's
Sure.
the
Yeah.
that's the challenge. But it's John, we're reporting on things, and it's just COVID has definitely changed the clinical trial landscape, and I don't know if it'll be forever, but something to keep an eye on, right?
So yes,
Yeah, do we want to go through all of these? I don't. I guess we
we
can.
can go with some of the highlights. Are very good. I mean, this is one that I honestly hadn't thought about. Is the actually for psychiatric disorders, it's become much tougher. I mean, you
Mm,
do realize. I mean, we've talked about how it's tough on people mentally. But for example, schizophrenia patients got tend to be more afraid of COVID, see their doctors less frequently, and maybe not be able to communicate as effectively through telemedicine, which has hampered diagnosis. Sage Therapeutics found that the change in dynamics of mental health also affected the way placebos performed in the control arms of clinical trials, which I honestly had no inkling of at all. So it's had an effect on clinical trials, but particularly for mental health disorders, it's actually had an effect on the patients in those clinical trials. So, that presents an additional challenge for the pharmaceutical companies. Just other things we can talk about.
Well, no, I think let's stop for that one just for a second because I think it's
Sure.
important.
Yep.
I think what we're seeing is not only is and because you're right, John, we hadn't even occurred to us. There's been a spike in depression and all sorts of psychiatric disorders due to COVID. That's really a known. I think they're still kind of on the rise. We don't know the full effect of this, the impact on, say, adolescents and children, right, or even some adults who, like my mom, for instance, still doesn't want to really leave the house, right? So I feel like that's got some degree of depression to it. And so now you factor in trying to do a clinical trial in a setting where the control arm is just off the charts. That's what's happening here. It's hard to predict that control arm. And then I think we're at a stage where I always thought it was kind of a tough space to be in to begin with because it relied upon a lot of questionnaire type things. There's no real measurement of someone's psychiatric state, right? So I think it's one that it was somewhat of an unmet need or undermet need, right? And now COVID's actually maybe set it back a little bit, and that's something where I think you'd see more investments in over time. I hope, that's kind of my take on it. But yeah.
So the other items are ones we can definitely just barrel through fairly rapidly. One was microbiome therapies need new COVID-19 screening, and this is basically more microbiome-derived therapies. They just want to be sure that the people involved in the trials don't have COVID too. Trial enrollment ground to a halt, and it's, again, we've talked about this, and this really impacted, for example, rare disease. We've already got a very small population to pull from, and they cite homology medicine, who had report in delays in enrollment for dose expansion phase of one of their trials, looking at a phenylketonuria, which is a rare inherited disorder that causes phenylalanine to build up in the body, and so it can be addressed with dietary restrictions, but it meant rather than people enrolling in the trial, they went back to the more tried and true approach of dietary restrictions, which isn't great for their life. It's not going to fix things for them.
Yeah, John, and moving along, right? COVID-19 becomes a new adverse event. I think everybody would understand what that one is. Like now it's something where you're trying to treat somebody and they might pop up with COVID. That wreaks havoc upon a double-blinded trial, right?
Yep, sure.
And then I think I'll do the next one. Site inspection delays stall new drug approvals. So the FDA it's all shut down. So the FDA can't get to facilities to approve them so that they can make drugs. That's a huge backlog.
And even with that, when it opened up post Delta, we thought we were going to see site inspections again. Then Omicron emerged and they shut down. So it's like, then they wait for that wave to disperse before they can start moving out. So they are moving forward in the US, but the overseas isn't happening.
Yeah, and a lot of our drugs are made overseas, right? A lot of the trial, a lot of the CMC work is done overseas, and the drug is imported in. So now, if you can't inspect that, you can't get the drug. I mean, just more things that we hadn't really thought about. And then certainly the last, I don't know if this is the last one, but supply chain. Good.
Yes. It really is the last one. Supply chain issues. I mean, everyone knows about that because it doesn't just affect pharma and us. It affects life because there's still supply chain issues across the board. I mean, I don't know what you're like for cat food, but I know there's a shortage of aluminium for cat food tins, and it's the one part of the supermarket where the shelves are still empty.
Ooh,
because
yeah, no, we have a lot of. That's interesting. It's not just cat food, but I didn't know there was a. I feel like there was a pet food shortage in general, and then we're still there's a lot of spaces on the shelves here that just aren't some certain items aren't coming back. I can't tell if it's just due to well, there are six other things, so the who needs a seventh version of cream cheese, for instance? I don't know, but there's still some things, which is interesting. So
yeah.
I did not know about the cat. I hope you I love animals, John, and I hope you can find the right food for
Oh,
yours.
we got them. I mean,
Good.
I'm a hoarder when it comes to that stuff. So
Yeah.
we're never going to run out, but certainly it's like it's more difficult to find
my
things. And
cat, my cat's a fat cat. We got her on some diet food. It's not working well
of
for her.
course, we have fussy cats, so you have to go for particular brands. So
Yeah, she's
of
she's
course,
kind of yeah, she's a little bit fussy, but she when she like what she likes, she eats a lot of.
yeah,
So we'll jump off of COVID now
yep,
and
yep.
hop into non-COVID stuff. John, why don't you tell us about the
So
first one?
this we touched on this because I think it had actually been authorized in Japan, but now there is some potential relief for contact lens wearers who suffer during allergy season, and that the FDA has approved J&J's allergy-fighting contact lens infused with antihistamine. So it's the first one on the market, and it basically it's Johnson and Johnson's Vision Care AccuView TheraVision lenses are laden with ketotifen, the same drug used in a variety of eye drops for taming reactions to pollen grass, pet dander, or dust. In clinical studies, the soft daily disposable contacts have been shown to work in as little as 3 minutes and provide itch relief for up to 12 hours. I mean, honestly, if you suffer from seasonal allergies, this is fantastic.
Yeah.
At least for me, I'm gonna be talking to my optician about this. But actually, I think it's great, but it's actually really interesting in terms of the challenges of actually doing this, because you've got two things that are working really two different goals with these lenses. You've got your lenses that are looking to correct your vision, and you've got a drug that is obviously looking to prevent itch. So the design had to be the way they had to incorporate the drug had to be done in such a way that it didn't interfere with the way the lenses operated. So I mean that's challenging. I
Oh,
mean,
it's amazing. It's amazing. Yes.
yes. I mean, so
Phenomenal.
what sounds simple, once you stop and think about it, it's really genius how what they've done. So yeah, it's
Is that see for me the moment you talk about the eye, it's immune privileged, and I immediately know it's not that easy, and there's
sure.
very hard to sample from it. But you're right. I mean, on the surface, it's like, oh, why can't they just take my contact lens and put a drug on it? But they talk about interference from other things, like you said, you're really trying to make sure that you got two things, right? You got to correct the vision and try to treat conjunctivitis and other
Mhm.
types of things. And so it's really phenomenal technology. It's part of the reason why I still wear glasses, John. I mean, I only had to wear them a couple of years ago, but I have no desire to get contacts because, man, my allergies as I'm getting older are horrible. I couldn't imagine what would happen. Even right now, my eyes are sort of watering. It's crazy time of year here in Kansas, and certainly, but my wife, on the other hand, I'm going to let her know about this because she is all in on contacts and would
Yeah,
love to have something that could
I,
treat it. Because she goes through a bottle of eye droppers like a week because her eyes are just.
I am too. I mean, well, as soon as I went to contact lenses, there's no way I would ever go back to glasses unless I can absolutely avoid it. But then I don't like putting things in my eyes, so I drop
No.
stuff I've never liked. So for me, it's almost like I'm the target audience on this
Uh,
one.
I'm
So
probably if my guys get anywhere, I can see without my glasses, but I use them because I don't want to like squint my eyes and give myself a headache and wrinkles.
mhm.
But if it gets worse, which I'm sure it will, I'll probably get surgery. Right? I don't know if you've had that, but I'm thinking that way. So, but the idea of having a contact that could help with allergies, on the other hand, that's almost attractive, right? Because my
Yep.
eyes are watering more. So good stuff, and it's approved. And hey, you'll get yourself some,
Yeah,
right?
talk to your optician. Moving on, this is interesting in terms of obesity may alter inflammation on a molecular level, throwing current treatments into question. And this was research done by a group at the Gladstone Institute, UCSF, and the Salk Institute for Biological Studies, reported that among obese mice, there were molecular changes in immune response to atopic dermatitis that likely affected their response to treatment. So they basically put some mice on a high-fat diet, others on lean, infected them, and then it said that the obese mice had significantly more inflammation and worse disease severity even after weight loss. Looking at the T cell response amongst the two cohorts, that the obese mice showed an unexpected and prominent response amongst T helper 17 cells. Um, and then it talks about, T helper 2 cells, etc. And then they also said they had additional detail from 59 human patients with atopic dermatitis, um, in addition to a longitudinal study of patients with asthma, that actually corroborated the findings from the mice, namely that obese patients were more likely to have indications of Th17 inflammation.
Yeah, no, I mean, if T helper 17 inflammation is somewhat of a newer pathway, John. I mean, in fact, I'm going to date myself. When I was in graduate school getting my immunology PhD, it didn't even exist. So that pathway has spawned in the last, like, I'd say 15 to 20 years, maybe more 20 years ish, right around. But anyhow, that this is still a fascinating pathway, one where these types of studies are going to further correlate, obesity and allergy and all of that may be kind of bound into one. And what they're showing is what you put in your mouth definitely has an effect on how you feel, right? Like you
Yeah,
are what
yeah,
you eat. And so the main culprits here are IL-4 and IL-13, which are they're really hallmark T helper 2 cytokines. And so this sort of 17 pathway is really kind of the way it kind of works is it sort of suppresses the good help and turns on the bad help
sure.
because you're combating your because of the obesity. It's kind of, and then TL17 is heavy in the allergy and inflammation space. So the two of them collectively have a negative effect on positive cytokines and produce more negative, and hence, when they go into these obese mice and they block IL-4 and IL-13, they restore the immune system back to more lean mice. So it's some of this is pretty well known, but I think the uniqueness here is they've demonstrated the obesity component along with the allergy component of it. So good stuff, and certainly more to come. And I guess for me, line me up. I'll take a T helper 17 shot if I could get one.
So it's actually interesting that they said there was one thing that they did. They added an anti-diabetes drug to treatment regimens and were able to stymie Th17 development and improve the response to anti-Th2 treatments. So basically, it sort of gives a potential pathway for combination therapies.
Yeah, and I think that is, you're right, John. That's sort of the new piece. They're correlating obesity to T helper 17 responses.
Yeah,
And
yeah.
that hadn't been elicited quite as well. Like I said, it's known to be the major role in allergy and inflammation. And then it's the other piece is it's more T cell driven, which is, again, that's the real root of things. To me, a lot of times we study stuff in plasma and serum, but not the cells, right? The cellular component is where those things in the plasma and serum are coming from. So if you can cut them off, you prevent the proteins from coming out. So anyhow, John, that's a good one. And then I think this is our last one, but
It is,
certainly not
yes,
the
yes.
last one. Last but not least, tell us a little bit about Intella, who we know a lot about.
So
Cool
yeah,
stuff.
so Intellia now has the data for number one unanswered gene editing question: does it last? So they have a, a, a therapy in development, and they showed data from a phase 1 trial. It's called NTLA 2001. Um, so the early data showed effectiveness based on the reaction of a biomarker, which is, I think it's called transthyretin. Sorry, hold on a tick. They they're
I just like it when you say, hold on a tick.
yeah,
Well, I can't get away with it like you do. No, it's the ATTR, right? The ML, the
I,
trans.
amyloidosis. It must be the condition, but they looked at, so the biomarker is transthyretin, which is reduced in serum, showing an effect. That's shown an effect of their of their therapy. Now, they've actually been monitoring patients longer term, because it's one of those things with gene therapy. We're still in the early days of it. How long is it lasting? And in the long term monitoring, what they found was that reduction at day 28 was actually reflected months later. So that, at least based on the biomarker, their conclusion is that their therapy is effective, because and so they haven't seen any increase in adverse effects. So it's basically doing its job in terms of suppressing the biomarker that increases with this condition.
Yeah, I mean, I think there have been several instances where one shot has been effective over the life, or at least with a lot of longevity. But I think that's not the appeal here. It's just more being able to monitor it, demonstrate it has long-lasting efficacy, but that doesn't mean you couldn't possibly get another shot 10, 15 years down the road, right? So I think this is really good stuff in terms of showing that we're at the forefront of gene therapy has been around a long time, but it's now become, we got like 20 approved cell and gene therapy products, over 2,000 clinical trials going on. All of that is like we're on this is like the space for us. It's a new frontier to some degree. And so these types of studies showing the ability to monitor the longevity of a drug product is paramount to seeing what's really effective and what's not. So great stuff.
Yes. So yeah, just to give a bit of background, is ATTR amyloidosis is a rare and fatal disease that occurs in people born with TTR gene mutations, which cause the liver to produce abnormal, often misfolded TTR proteins. Damaged proteins, which in a healthy form help carry thyroid hormone and vitamin A in the blood, build up in the body, causing a host of problems in the nervous system, heart, and other organs. So that's what it's to address.
Don't mess with vitamin A, John, right? That's why they tell pregnant women to stay away from it, right? And now you see it here in these genetic disease, right?
Yep.
Good stuff. And they're reduced in size, right? All these things are happening to these individuals. But interesting, John. So good stuff. I think that wraps up the news, unless you had anything else.
Nope, not at all.
Well, let's just jump right into it, John, right? What an exciting main topic we have today. Building for the future, new investments here at KCAS. We have lots of new technologies. Let's start there, John, right? We're launching a whole tirade of technologies. I'll go with what we're working on the, let's say, molecular side, right? We've within the last month, we've started. Sorry, John, go ahead.
No, I was just clearing my throat. I think mostly the most of it is on the molecular side. I think we've talked about that we've been building up to this. So obviously, around this time last year, we broke ground on the new building, which we'll touch on, and also in parallel with that, we announced that we had new majority ownership. Who were obviously looking to help KCS grow. So part of that growth that we're looking at is this investment in new technologies to take us into new and different directions, or expand current directions to help us more successfully address problems that customers have. So I mean, a lot of it is. I'll let you mainly touch on the new pieces, but say in the existing side of things, on the mass spec side, there's always that push for sensitivity. It's like so we have a variety of mass spec platforms, and as the technologies improved, the sensitivities got better, and as a result, we've ended up with API SciEx API 4000s, 5000s, 6500s, and with each of those iterations, the sensitivity of the instrument, and the ability to get lower LOQs or limit of quantitation, and see lower amounts of drug in biological fluids has improved. So, in common with many labs, we stuck with SciEx, and recently, so another part of this, as we've moved into the hybrid LCMS sphere, where we're looking at large molecules, but to quantitate large molecules, that's almost the area where we're seeing the most demand to get even more sensitive than we're already able to do. And so we recently bought two Cyax API 7500s, which is their most recent triple quadrupole mass spec, where again, it's what can we buy relative to what was previously state of the art, how much sensitivity can you gain, and from that sensitivity, how much more can you drill into what's going on with these biological drugs, and provide answers, and so that for us is a big, hopefully a big step forward. We've done some head-to-head tests between the 6500 and the 7500, and we've seen gains. So it's then how much further can we take this. And another piece of it is, if you've got that additional sensitivity in hand, it makes your development easier because you've got that ability to go lower, and it makes achieving those levels that the clients are asking us to hit that bit easier. So that's really the big investment in the mass spectrometry side of things at the moment. I mean, obviously we're looking at other technologies for the longer term, but right now that's where we stand on the mass spec. And then moving into your side of the business, you have a lot more to talk about.
Yeah, no. Well, I mean, I think you're right. I kind of jumped right in, but over a year ago we broke ground on a new building, and we're very proud of the fact that through the COVID supply chain, we're on time and on budget. We're due to open our building, or at least start transitioning into the building end of this month, end of April, early May, and we have the good fortune of it being in close proximity to our current facility. The current facility we can keep open until at least the end of the year, so we're going to take a couple months to move. We have a very strategic plan, but through that move, John, that's really where we're going to up our technologies, right?
Yep.
And so you've already talked about the 7500s. We have a couple of them in-house, but you mentioned 4000s. We're going to continue to swap those out and gain the gains in the 7500 space. And it's not surprising that it just gets you there faster is what it does. It takes less development time to get a more sensitive method because we got a better back-end instrument from a 6500 to a 7500. And so jumping into the other new technologies we have, and we've got this is why we have a 3-year plan, John. We've got a lot of strategic planning. We are a process-driven company. We're a solutions-based company. And so we have in an incubator space brought on already digital drop PCR, qPCR, and ELISpot technologies. And so if you're not familiar with the first two, those are molecular platforms that allow you to measure, PCR, and then through some reverse transcriptase, you can do mRNA and things like that, but you can measure all sorts of genetic material using standard PCR equipment.
So what does can you break that down further? What does that mean to the layman? So what do you mean by PCR, and giving, what kind of data did you get from that?
Well, I think it's a wide range. First, you can look for genomic material that's part of a drug. So, for example, if you were to take a cell and insert a piece of genetic material like a CAR T cell, you can track that genetic material. That becomes a PCR test. Also, if you're using any sort of phage displays or phages or things of that nature that have some genetic material to it, you can measure. So it has some PK applications to it, and that's an emerging thing, is validating GLP validations of PCR assays is still something that is they have performance characteristics, but it's still evolving. And then the other piece is biomarkers. And so every cell has genetic material in it. You can start looking at, is it expressing a particular, like PD-1, checkpoint inhibitors. Those are big biomarkers for genomic testing, John. And then you can start looking at tissues, or even like a lot of therapies will knock in a protein, so you can look at it on a genetic basis. The platform for PCR is, and this isn't new, right? This is something that people who have been doing PCR reactions for close to 30 or 40 years now. Yeah. I hope I hope I got to what you were going for there.
Yeah, I think
the primary purpose for the primary purpose for PCR reactions in a GLP bioanalytical lab is to do PK analysis, and then the other is biomarker analysis, and in particular probably like things like tissues and lysing of cells. So it's like that non-liquid matrix type thing. There are some circulating DNA, And that's another place. So you can look at DNA in a plasma sample or a serum sample, but really it's about busting open a cell or busting open a tissue and looking for something is either high and you're trying to bring down, or is low and you're trying to turn on, right? So very powerful tools, very sensitive tools. And then the digital drop PCR. So this is phenomenal. It allows our system can do 20,000 droplets at a time. So you can actually take 20,000 assays in a single drop, John. That's how that works. And I'm still learning a little bit more about this technology, but we got some wonderful scientists that gave me a little bit of a education when we were at the building this past week. And so within that, they can multiplex in a variety of ways, because there's really just two fluorescent probes. But if I got 20,000 wells, I can stick a whole bunch of different probes all on the same. And it's like a Luminex system. They change the fluorescence a little, and they can, you can do a multiplex in a wide variety of ways because of those 20,000 droplets in there. And that's the power of it. It doesn't require a lot of material. It's just an incredibly sensitive method. But really, it's, most of the work we can do can fall on a qPCR instrument.
Sure.
Yeah. Instrumentation. And so those two instruments, we have fully functional and operational. And then as we move into our new building, we'll have them GLP, which is not an easy task. And we certainly feel like we pride ourselves on our quality, and we're going to make sure we're experts at it before we start offering that service. So it'll be sometime towards the end of 2022 before we can do full GLP, probably into 2023. But today we're open for business to do feasibility studies. All that equipment will be moved into our fit for purpose built unilateral flow. So the key piece, we've built a big chunk of our laboratory where the sample will go into one entry point, and it comes in kind of dirty. And then as it makes its way, there's pass through. It never goes into the hall, just pass through windows. And it gets cleaner. And then to some point in the middle of the lab is where it's the cleanest. And then it is going to go into the instrumentation space, and it starts to get dirtier. So it has this pathway of protection to it. It's high quality filtered air. It's a unilateral workflow. And I mean, it's just huge and fabulous, and I'm so excited to offer the services to help with global health, right? I mean, this is an amazing thing to have right next door, right? The other piece is it's right next door to our liquid binding assay. Sorry, go ahead.
If I can just interrupt very briefly, that's an important piece. I mean, you say we obviously we have a strategy of where we want to be in over time, but part of what the driver of this was listening to what customers wanted, because we were hearing more and more that people were looking for this service, that we, as the building's been designed, this became part of our strategic plan to include in that building, such that we could offer it and have it as a service going forward. It was all very much listening to our customer base.
Yeah, and John, I think the power of all of this is it's right next door to a massive ligand binding assay lab, a massive biopharma LCMS lab, and a massive pharma lab. It's all, and we got flow cytometry and cell culture suites. I can't wait to open the building, right? 70,000 square foot, and man, is it mostly lab. We are incredibly frugal with how we've built this building. It's amazing. It's, again, fit for purpose. And hopefully anybody listening, come visit us. And then John, it's actually, I think we're going to do, because I feel like we're going to get one of our molecular experts on here to describe more of that instrumentation.
Sure. Yep.
And to round out the molecular, we are bringing on next gen sequencing, but we're staging things. We don't want to overwhelm ourselves. We certainly don't want to let down any of our existing clients. So that will be more in the 2023. We'll bring on next gen sequencing.
So so you touched on the ELISpot. Where does that come into things?
Yeah, so there are two gold standards for measuring augmentation of immune cells. One is flow cytometry, and the other is the ELISpot. And what do I mean by that? So you can take a person's sample, blood, predominantly you isolate out the peripheral blood mononuclear cells, and you can look at that individual and say they have X number of T cells, in particular T cells, that are reactive against whatever it might be, a tumor antigen, a virus, all sorts of things you can look at. And you can say, okay, before I vaccinate or treat this individual, they have X number of cells. It's something like maybe 5 per 5,000 or something like that. And then you give them either a drug, or a vaccine, and then you measure them a couple weeks later or a month, and maybe you dose them again. But that number is going to steadily increase from 5 that are specific to a given protein to 5,000. And how that works is it's all based off of you count T cells that are producing a cytokine of interest that you're trying to alter the immune system to make. So, for instance, I might have right now probably a relatively high number of like CMV or EBV, Epstein-Barr virus. We all kind of have that. T cells. We might have maybe 100, but if you gave me an EBV vaccine, I'm going to go up to like 500 versus say 100. It's going to go like log scales higher or tenfold higher. But in a tumor, like in immuno-oncology setting, if you know what MUC1 is or EGF, these are breast cancer tumor antigens. Somebody who's sick and whose immune system has failed them, they might have only a couple or maybe none. Or they might actually have mucin-specific T cells that make things like IL-4, so that T helper 2 we're talking about. So that's when someone's immune system is exhausted, and chronic inflammation leads to exhaustion, which leads to T cells to be reprogrammed and make the wrong cytokine. So now you're trying to augment someone's immune system to make the right ones against the tumor antigen of interest. That's how you treat immuno-oncology patients. So ELISpots and flow cytometry have the ability to measure those with a specific number of T cells against a specific epitope. And so the ELISpot is a plate-based method. And what you do is it's got a nitrocellulose membrane. And on that membrane, you put the cytokine of interest. And we can do up to 4 colors at a time. We have fluorescent spots. So we can do 4 different. So we can measure like IFN gamma, TNF alpha, IL-4, and say like IL-13. Those 2 of them are T helper 1, 2 of them are T helper 2. That type of measurement allows you to measure the efficacy of your immuno-oncology and actually has a lot of vaccine application to it. And,
sure.
So those, I hope I'm doing that justice. Flow cytometry uses, that's a tube-based. You put it on a sip or it sucks up the cells, and through hydrodynamic focusing, they put this through some sheath fluid, and then one cell at a time, you put a laser through it. So both of them have different principles to it. Some people believe one's better than the other. I'm not going down that road. Just what I'm saying is those are the 2 major ways to measure if you're augmenting an individual's immune cells. And it's a gold standard for immuno-oncology and vaccines. And we've been doing flow cytometry for, I don't know, better part of, say, 8 or 9 years. And of course, we acquired Flowmetric.
Sure. Yeah.
We feel like we're the world's experts in that. Bring it on the ELISpot platform. It's near and dear to me, John. If you can't tell, I've been doing flow cytometry and ELISpots for a long, long time. I've supported a number of clinical trials, a number of viruses and vaccines that we've mentioned on this show. I helped get approval on. And again, I can't talk too much about it, but the companies I've worked at have helped that. And so we're very excited, and we have personnel that know it well. We're very excited to bring it on. It really does round out the main thing it tests is pharmacodynamic and biomarkers, as well as safety. It has a lot of safety applications to make sure you're not perturbing one's immune system. Remember, you're trying to augment the immune system. Sometimes you want to make sure you're not disrupting their, CMV responses, or not disrupting their pneumococcal responses, if that made sense, John, right? Sure. Yeah, if you augment someone's in the wrong way, they're going to stop producing. So there's some safety issues. And now I'm babbling, but go ahead, John.
No, that's really, really helpful. Shall we move on to actually the new, we've touched on the new building. Shall we talk about the new space? Or do you have any more to say on this piece?
No, I think I covered our new platforms. We're going to bring on something called the Ella, which is a Western blot-like instrument for biomarker analysis. That's another cool little expansion of our services. But just to recap, it's qPCR, digital drop PCR, ELISpots, next-gen sequencing, and the Ella, all coming to you from KCAS. So yeah, it's great stuff.
It's a big deal because we talk about the new building, and there's a couple of things I want to touch on that. I mean, so a big part of it is it's more space. So that extra space gives us the ability to expand. You're right. We're talking going from around 30 odd thousand square feet to just shy of 70,000. But the other key part to it, the building, we have the ability to expand the building beyond its current footprint, which could take us up to 100,000 square feet, depending on need. But the way we're doing the move, because the move doesn't have to be done quickly, we are actually going into the new building with new instruments, so we can get new instruments set up, ready to move, and then we do a phase move of other of the groups, one at a time, to again get them get them set up before we then turn to the next group to move them over. It'll be a case of, certainly for the mass specs, and I've been through this previously in another role, where what we did, and KCS is taking the same approach, where you have an assay that runs on an instrument. You basically show how that works on the instrument before the move. Then we will basically obviously wind the instrument down, because you're taking back up to atmospheric pressure, etc., from vacuum. You're switching it off. You transport it, then you set it up in the new facility, pump it back down, and then we'll rerun that assay to show that the performance is equivalent between the two facilities. And at that point, then you're going to judge the instrument fit for use for moving forward. Another key part of it is we're still retaining the KCS approach of keeping all our services under one roof, so it still maintains that ability that we've talked about in previous podcasts. If we've got a problem using one technology, we still retain that ability to flip to another technology that may be able to provide an alternative solution quicker. That's retained, but we're still taking that approach. Another key part of a bioanalytical facility is how you look after your samples. So we have numerous freezers full of samples, because you're maintaining their what's the word, their integrity. So a great way to move between facilities for frozen samples is to actually, we're renting trucks where you can wheel the freezers out, plug them in, and so the freezers will actually be running on the truck between facilities before they then get put into the new facility. So we've got a lot of this is going on. It's like you say, it's a well planned move, and it's because we've got that overlap of facilities, we can take as long as we need to make sure that it goes properly.
Yeah, John, it's beautiful. You did it, like, I love it. You're as excited as I am. I can feel it. I think
Oh,
that's about as excited as a British guy or Scottish guy gets. I could feel it coming out of you.
yeah. Well, I mean, I've been through this before. It's. I mean, I obviously I thoroughly enjoy my job and I thoroughly enjoy the company, but to move to a new custom built facility has a transformative effect on how you operate. It sort of it sets off something within people. So it definitely boosts how people feel, but also it boosts people's reaction to you. You're right. You want people to come and visit the facility because they will be blown away, and it will change the perception of the company out on the street.
Yeah.
I mean, I was on obviously my trip round. I went out with Dan to visit some customers last week, and because of being in Kansas recently, I was able to share photos of the building from the outside to customers and saying that's where we'll be in like a month and a half's time, so come and visit, because it's something impressive for people to get a handle on.
Yeah, John, I mean, like you, I love my job, and I'm so excited for the new building. I've been here 9 years. I almost want to say I can't believe it's taken this long, but the reality is we have been tactical, precise, and it's amazing to watch us in action. We're so blessed to have so many people that have done this before, so it will go smoothly because of the planning, right? And you
Sure.
touched on some of that. We're not actually going to officially cut the ribbon on this until 3 months after, so May, June, July, end of July, August timeframe. Probably August is when we're going to say it's officially opened. And the beauty of that is we have our current facility well through the end of the year. And if possible, maybe even longer if needed, but we don't anticipate that. And then you talked about the redundancies of instruments, and you're right. We're going to put in a bunch of new mass specs, but on the LBA side, we're basically just going to double our footprint. We're going to put all new MSD instruments, and I think we got a Luminex instrument coming, and a bunch of plate washers, and a lot, plate readers and everything, and that we'll bring those online, and then we'll take all the equipment in the new building, move it over. So it's a staged approach, and then boom, we're going to be like double or almost triple our capacity in the new building in the LBA space. In the other piece, John, one of the things you talked about, the expansion from 70,000 to 100,000. John, our growth is so exponential that there, as you know, that's a done deal. We will expand probably
Sure.
quicker than we ever imagined. So we had thought maybe we could fill that building. It might take us upwards of a couple years. Well, we're projected to really outgrow the space if we continue to hire, and we're going to talk about that in a minute because that's a key piece of this. We think we're going to need that space almost immediately, John. We're going to start building probably sometime in 2023. So great stuff there. John, I don't know, before we move off, I think I touched on our cell and gene therapy investments, all the molecular stuff, those LE spots, that's predominantly in that space. So before we move on to our next topic, John, any closing remarks on the first few?
No, I think feel free to go ahead.
Sure. Yeah, so I kind of just touched on expansion of the workforce. John, we've talked about this. We recruit like we're a Division I football team. All of us are recruiters, and we do it. I'm so proud of the way we do it. Meaning, we seek out individuals. We want individuals who are running towards us, not running away from something. We do extensive hiring processes. We've got a whole team out there for that. It's great. I get out in the local community here. I also try to go out nationally. We'll recruit from anywhere. But the local stuff that we're doing, it's great. We're getting into some community colleges, and we have one of the largest community colleges in the country. It's called Johnson County Community College, and we'll do some internship programs with, what are basically first set. That's an associate degree level, but they're predominantly the first 2 years of college, and then we have KU and K-State and all that. And we're getting very much in the latter stages, and then there's an applied biotech degree at a KU that I actively teach in, and so those are all some of our organic growth there. And then really we have been very good at going and recruiting top-notch scientists, senior scientists, principal investigators from across the nation, both coasts. And really, due to some of what's happening, Kansas City has become a very attractive place, a very affordable place, a very safe place. So it hasn't been that difficult, but like anything, hard work and luck go hand in hand. And so, John, I don't know what you want to say about our hiring, but we've got, I think, 4 or 5 people dedicated to recruiting, because we have 30 open positions right now, John, right? And where our number is creeping up daily, it's hard to keep up with. It's hard to keep our capability slide deck up. You pull it open right now, it says 215 employees, and that was updated about a month ago, and we've got like 230 employees now, right? So it's challenging to keep up with. And I'll say one last thing. I used to know everybody by name. Man, that has been very hard to keep up with. I think I know probably 8 out of 10, maybe 9 out of 10. But every time I walk in the building, there's a new face, and I try to get to know them.
It's even tougher as a remote employee. You walk in, and again, there's an awful lot of people I have no idea who they are, because obviously I'm only there occasionally. I think, it is. It's great that we, like you've mentioned, on the having an internal team dedicated on recruiting, because I'm regularly being contacted by people I know saying, "Oh, just being contacted by KCS. What do you know?" And again, you're right. It's because I enjoy the company, because I love my job. I'm motivated to sort of say it's a great place to work. Please.
Yeah, we have some experts, John, right? World-class experts, people who have helped. They've worked in pharma, they've worked in big biotechs. For whatever reason, they're here in Kansas City now, and that is really causing what you're feeling. People are beginning to reach out to me. I have some friends from graduate school and other places saying, "What are you guys doing over there? I keep seeing you."
Yeah. It's interesting. I think it's again, being out on the road last week. I'm trying to think where it was, but someone said, Yeah, I'm seeing your name everywhere in terms of from where we were a few years ago. Certainly prior to me joining, I didn't make a difference. This is just the way the company's gone. I remember when I was first approached, I actually contacted others of my industry contacts to say, what do you know about KCS? Because honestly, my knowledge was not there, but over the last few years, we've become much more of a presence out there, and people.
Yeah, my favorite phone calls are from people I know that work at competitive labs. That's the best call I get right there.
Sure. Yeah,
No. Well, John, I think we covered most things. I don't know what else we have. We talked about how well, certainly one of the biggest things that we wanted to keep was that we're all under one roof. We touched on it earlier, but that ability, and it's palpable, John, right? How many people are investing in feasibility studies using both platforms, meaning someone will do biopharma, or what we call hybrid mass spec, along with an LBA, and that's happening at a much higher rate for a few reasons. One, we're getting more efficient at it. And then it actually answers the question right up front. You don't have to go to any regulatory bodies and say, oh, could you do a more sensitive method over here?
yeah,
No, we
yeah,
already know. We came out of the gate strong and we
yeah.
worked hard to produce one. And really it's, I know there's from my seat on the bus, it's still ligand binding assays and using hybrid mass spec. It's still like, it's kind of equal, John, right? They're still both, some projects work better on LBA, some work better on biopharma. I feel like we did about like 10 or maybe 8 or 10 of these last year, and maybe some of them this year, and it's like 50-50, right? But
Sure.
that ability, and what happens there is we can, I was like you traveling last week. I was out in the field, and we had a number of people talk about, well, it's just so hard to pivot from one platform to the next. And that's when I said, not at KCAS. We can do it. We have a number of examples where we'll do it within a week, especially if, we pride ourselves on not exhausting all of your days on one platform. So if there's any days left, we can just start immediately moving. And mind you, the reagents are all there. We don't have to ship it to another site. That was one of the key things we wanted to keep as a really an efficiency and just a quality of the product from KCAS.
Sure. Yeah, absolutely. Yes. So it's. I hope it comes across. It's exciting times now and exciting times ahead.
Yeah, and we're excited, but really it's going to impact our clients. It's going to impact just in general. I hope I don't sound arrogant, but we're helping global health, and
Sure.
really when we go from 33,000 and we're doing hundreds of maybe 300 or 400 PK validations across all of our platforms, that number is now going to go even higher. Probably got our capacity to, like I said, double or triple, and that just completely increases the as I see it, global drug development, because we've got a lab in Milan now, and we definitely have a global footprint, and more to come on what we'll be doing at KCS there.
So yeah, so let's stop there. I think we've covered the main topic. We really want to convey what's going on at KCS because, like I say, it's exciting times right now. So we've done that. What's your weekend plans?
Yeah, John, this is full on the cross season for me. And if you're not aware, spring in Kansas City is not nearly as warm as the summer. And so even today we've got, I don't know, a dusting of snow fell. It's crazy times out here. So what I mean by the cross is my son is a sophomore, so he plays on the JV team and he plays on the varsity team. So there are 2 games even this evening that I'll be sitting in, 40-degree weather in the stands watching lacrosse. I often get on the field and help participate by keeping, I do the book or keep score or something like that. But then he has a game on Sunday, and this week's Palm Sunday and everything. So that becomes the focal point of the weekend. So I'll spend a lot of time outside in the cold. But with that, I always plan for some food, John. I did pick up some pizza dough. I can't go a podcast without talking about pizza. I haven't been able to use my uni because the temperature hasn't gotten up, and then the wind, but I have really refined my game inside making pizza, and I go and get the pizza dough from a place called Grinders. And I don't know, every time I make it, my son and wife look at me and they're like, it's just better and better. And I've gone out and ground up some mozzarella. I'm getting a little more sophisticated. I don't just buy a bag of mozzarella. I'm getting good mozzarella. I've made some pasta sauce. I still got probably 5 or 6 jars of that, quarts of pasta sauce because I made a big batch a month ago. And so, and then I've been getting some high-end like Asiagos and kind of mixing it in with it. And then, of course, I'll drive 20 minutes to pick up pepperoni. So, John, I don't know. It seems somewhat boring. I'll do some yard work, hopefully, because there's a lot of trees, but I don't have anything glamorous going on this weekend, John, is what I'm telling you.
So I only have an abbreviated weekend this weekend. So on Saturday, it's obviously we talked a while ago about my daughter got engaged. So we're actually going to look at a potential venue. We're aiming now for her wedding in 2023. So this is the first step towards actually formalizing things. So we're going to visit that tomorrow.
That's exciting, though. That's
Yeah,
exciting.
absolutely. No, it's great. After that, I'm flying west on Sunday, ready to be back on the road again. So it's really great to be traveling and meeting people and talking business. So
Yeah.
I'll be doing that and going on the road with Amy, Amy Mize, who we've had on the podcast, and Adrian Chicos. So that's next week, but then I'm actually on the road again the following week. So I'm into one of those modes where I seem to be traveling a lot. We've got our scientific advisory board meeting early in the week in Kansas City, so I'll be over for that. But almost more importantly, we have a webinar on April 22nd to talk about hybrid LCMS MS, and well, obviously I'm going to be the introduction to it, but Dawn Duffield will obviously carry the meat of the webinar as the expert in the field. So very much looking forward to that.
Exciting. Go ahead. No, go ahead.
Yeah, so that's really it. Yep, it's back into travel mode in terms of but also because things are slowly starting to move, your mind starts churning as to what you want to do around the house and the garden, etc. We just got daffodils coming out. I'm still, it's a constant battle with deer, so I've got a bunch of things covering tulips to make sure they survive this year rather than all get eaten. So, and then there's a number of things that we're going to be doing that will change how we spend our time over the summer. But yeah, we got plenty on at the moment.
Yeah, no, and just following along, I traveled last week. We got AAPS NBC coming up at Anaheim. That's like May 9th through like the 13th. We'll be exhibiting there. So I'll probably make a trip out to Anaheim if my schedule permits, which I hope it will. I'm trying to move some stuff around. Not that I'm that busy of a guy, but really I do volunteer quite a bit for my son's lacrosse team, so I don't want to let them down. And then, John, we got Easter coming up. That's probably a big one for me. Excited to celebrate Easter in a I shouldn't say non-COVID, but at least in a more ability to visit with family.
Sure. Yeah, absolutely.
Yeah. And John, baseball started. It was opening day at the Kauffman at the K. Royals opened up yesterday. Fortunately, I didn't even see if they won or lost. And then, of course, I don't know, I'll throw in a soccer plug. Man, did Chelsea look bad midweek against Real Madrid. I don't know if you saw that.
I did not see it.
They got thumped. Man, they got thumped 3-1 on the first leg. It leaves them on up. They give up 3 home goals, which is terrible. They got to go on the road now and win like 3-0 or 3-1, and that leads to shootout. It's a big ask against a very good Real Madrid team. Benzema had a hat trick. It was
to be fair, Real Madrid are a bit up and down. It depends what day you get them on.
I know. I don't know. They had a fluke goal, but other than that, Benzema just won the game single-handedly. Yeah. So anyhow, and then of course the Premier League race is amazing. Man City and Liverpool are gonna duke it out. At one point, it looked like Man City was going to run away with it. That they've been reeled back in, and Liverpool's right on their tail. So anyhow, and then I know I won't be able to make the Sporting KC game. They're in town, but like I said, I got a lacrosse game that will be at the same time, so I won't be going out to see them. But I want to get out and see Sporting, who are struggling a little bit, actually. They need me to get out there, John.
I'm surprised you didn't touch on the Kansas winning the NCAA.
Oh, hey! Yeah, it's in part because I was traveling when it happened, but that's huge. What a fun time. What a great time for the Kansas City region here. And I don't know if you saw the Lawrence, where KU is. There was so many people out. And then, for having a 16-year-old during this time, it was just a blast. They didn't give him homework that night. They kind of took it easy on him on Tuesday after they won. He kind of was out trying to find a shirt at like 11 at night. Just exciting things, you know.
Yeah.
It was good to see the youth. They were running around. They were having a blast.
I'll be honest. I wanted UNC to win it, but yeah, hey ho.
Yeah.
Next year's a whole other tournament.
No, and KU was probably the best team in the tournament. It doesn't always
Sure.
work out that way. They looked pretty dang good. So
Yeah.
with that, John, I'll see you in the funny pages, and have a great week, and we'll connect real soon.
Absolutely. Yep. Thank you very much. Have a good one.